Chemical digestion in the small intestine is a complex and highly coordinated process that breaks down macronutrients into their absorbable components. This process involves multiple enzymes from different sources working together to ensure efficient nutrient absorption. Understanding these mechanisms is essential for comprehending both normal digestive physiology and various clinical disorders that can disrupt this vital function. Digestive enzymes in the small intestine come from two primary sources. The pancreas secretes powerful enzymes into the duodenum, including proteases, lipases, and amylases that break down proteins, fats, and carbohydrates. Additionally, the brush border of the intestinal epithelium produces its own enzymes that complete the final stages of digestion. This dual-source system ensures thorough and efficient breakdown of nutrients. The pancreas plays a crucial role by secreting sodium bicarbonate to neutralize stomach acid, creating an optimal pH environment for enzyme activity. Pancreatic enzymes include trypsin and chymotrypsin for protein digestion, pancreatic lipase for fat breakdown, and pancreatic amylase for carbohydrate processing. These enzymes are secreted in inactive forms and activated in the intestinal lumen to prevent self-digestion of pancreatic tissue. Brush border enzymes are located on the microvilli of intestinal epithelial cells and perform the final steps of digestion. These include disaccharidases like maltase, sucrase, and lactase that break down sugars, as well as peptidases that complete protein digestion. Deficiencies in brush border enzymes can lead to malabsorption syndromes, with lactose intolerance being the most common example, affecting a significant portion of the global population. The breakdown of macronutrients requires precise coordination between pancreatic and brush border enzymes. Carbohydrates are broken down into monosaccharides, proteins into amino acids and small peptides, and fats into fatty acids and monoglycerides. This coordinated process ensures that nutrients are reduced to their smallest absorbable units, ready for transport across the intestinal epithelium into the bloodstream. Bile, produced by the liver and stored in the gallbladder, is essential for fat digestion. While bile does not contain enzymes, it emulsifies large fat globules into smaller droplets, dramatically increasing the surface area available for lipase action. This emulsification process is critical because lipases can only work at the water-fat interface, and without bile, fat digestion would be severely impaired. Bile salts undergo enterohepatic circulation, a highly efficient recycling system. After aiding in fat digestion in the small intestine, approximately 95% of bile salts are reabsorbed in the terminal ileum and returned to the liver via the portal circulation. This recycling process allows the body to maintain an adequate bile salt pool with minimal daily synthesis, conserving energy and resources. When bile recycling is disrupted, such as in ileal disease or after ileal resection, significant clinical consequences can occur. The loss of bile salts leads to fat malabsorption and steatorrhea, as insufficient bile is available for proper emulsification. The liver attempts to compensate by increasing bile salt synthesis, but this may not fully replace the lost bile salts, resulting in chronic digestive problems and potential deficiencies in fat-soluble vitamins. Understanding enzyme-substrate relationships is fundamental to comprehending digestive physiology. Each enzyme is highly specific for its substrate. Amylases break down starches, proteases target peptide bonds in proteins, and lipases cleave ester bonds in fats. This specificity ensures efficient and controlled digestion, with each enzyme contributing to the breakdown of specific macronutrient types without interfering with other digestive processes. Carbohydrate digestion begins in the mouth with salivary amylase and continues in the small intestine with pancreatic amylase, which breaks down starch into maltose and other disaccharides. Brush border enzymes then complete the process by converting these disaccharides into monosaccharides like glucose, fructose, and galactose. These simple sugars can then be absorbed through specific transporters in the intestinal epithelium. Protein digestion is a multi-phase process beginning in the stomach with pepsin and continuing in the small intestine. Pancreatic proteases, including trypsin, chymotrypsin, and carboxypeptidase, break proteins into smaller peptides. Brush border peptidases then complete the digestion, producing free amino acids and small di- and tripeptides that can be absorbed. This sequential breakdown ensures complete protein digestion and optimal amino acid availability for the body. Fat digestion is particularly complex and occurs in several stages. First, mechanical churning in the stomach and bile salt action in the small intestine emulsify large fat droplets into smaller particles. Pancreatic lipase then hydrolyzes triglycerides into fatty acids and monoglycerides. These products combine with bile salts to form micelles, which are essential for transporting lipids to the intestinal surface for absorption. This multi-step process is critical for the absorption of dietary fats and fat-soluble vitamins. Phospholipids and cholesterol require special digestive considerations. Phospholipase A2 from the pancreas breaks down dietary phospholipids, while cholesterol esterase hydrolyzes cholesterol esters into free cholesterol and fatty acids. These specialized enzymes ensure that all types of dietary lipids, not just simple triglycerides, can be properly digested and absorbed. The secretion of digestive enzymes is tightly regulated through multiple phases. The cephalic phase begins with the sight and smell of food. The gastric phase responds to stomach distension and chemical stimuli, and the intestinal phase is triggered by chyme entering the small intestine. This coordinated regulation ensures that enzymes are secreted at the right time and in appropriate amounts to match the digestive load. Several key hormones coordinate digestive enzyme secretion. Secretin stimulates pancreatic bicarbonate secretion to neutralize acid. Cholecystokinin, or CCK, triggers pancreatic enzyme release and gallbladder contraction. Gastric inhibitory peptide slows gastric emptying to prevent overwhelming the small intestine. These hormones work together to create an optimal digestive environment and ensure proper nutrient processing. Clinical disorders of pancreatic function can severely impact digestion. Acute pancreatitis involves inflammation that can damage enzyme-producing cells. Chronic pancreatitis leads to progressive loss of pancreatic function, requiring enzyme replacement therapy. Patients with pancreatic insufficiency must take supplemental enzymes with meals to ensure adequate nutrient digestion and prevent malnutrition. Celiac disease is an important malabsorption syndrome caused by an immune reaction to gluten. This autoimmune condition damages the intestinal villi and brush border, leading to decreased enzyme production and impaired nutrient absorption. Patients experience symptoms including diarrhea, weight loss, and nutritional deficiencies. The only effective treatment is strict adherence to a gluten-free diet, which allows the intestinal lining to heal and digestive function to recover. Lactose intolerance is the most common brush border enzyme deficiency worldwide. It results from decreased production of lactase, the enzyme that breaks down lactose, the sugar found in milk. This can be primary, developing with age, or secondary, resulting from intestinal damage. Symptoms include bloating, gas, and diarrhea after consuming dairy products. Management involves dietary modification, lactose-free products, or lactase enzyme supplements. The integration of chemical digestion processes in the small intestine demonstrates remarkable physiological coordination. Multiple enzyme systems work simultaneously on different macronutrients, while hormonal signals ensure proper timing and quantity of secretions. The brush border provides a final checkpoint for digestion, and the enterohepatic circulation of bile salts exemplifies the body's efficiency. Understanding this integration is essential for recognizing how disruptions at any point can lead to clinical manifestations, and for developing appropriate therapeutic interventions. This comprehensive system ensures that the nutrients we consume are effectively broken down and made available for absorption and use throughout the body. In summary, chemical digestion in the small intestine represents one of the most sophisticated and coordinated physiological processes in the human body. From the dual enzyme sources of pancreas and brush border, to the critical role of bile in fat emulsification, to the hormonal regulation that orchestrates the entire process, every component works in harmony. Clinical disorders affecting any aspect of this system underscore its importance and complexity. A thorough understanding of these mechanisms provides the foundation for comprehending both normal digestive health and the pathophysiology of gastrointestinal diseases.